Please go ahead.
Okay. So, well, let me get started. I want to thank Mrignani for asking me to serve as the host of today's webinar. So this is a series of webinars that's being sponsored by O2M Technologies. And so we'll have a presentation by Professor Benoit Driesschaert. Can you correct me if I don't have it quite right?
Yeah, Driesschaert. That's fine.
Driesschaert. Okay.
Yeah.
And I'll give a little bit more of an introduction on his work in a minute, but let me just sort of go through the introductory slides that Mrignani has provided me. So this is, I believe, going to be the next seminar by Professor Gareth Eaton from University of Denver, and be sure to sign up for that. That will be, the time is different, as you can see, it's from 2 to 3. And then the June 16th seminar is actually, the other one was July, right? Yeah. So the June 16th seminar is by Joshua Biller, and that's also at 2 p.m. And so again, be sure to sign up for that. Let me give a brief introduction for Professor Benoit Driesschaert.
Driesschaert. I mean, I know.
Driesschaert. It's a CH, hard CH. Okay, Driesschaert. So Professor Driesschaert, he did his training. Most of his educational training is in Belgium. He got his bachelor's degree from Catholic University of Louvain, Belgium, in 2003. He then obtained a master's degree in organic chemistry and in biophysics, and stayed on for a PhD, which he received in 2013, and continued to work at the institution for a little bit longer as a postdoctoral fellow, and then moved to the Ohio State University in 2015 as a postdoc, and I think really got involved in a lot of this triddle work at that point. He started his independent position at West Virginia University in Morgantown, West Virginia, and in 2018, and has been working on the synthesis and study of various derivatives of these types of persistent triddle radicals for biomedical applications, especially imaging type applications, including oxygen imaging. He has really laid out the details of the synthesis of the types of triddle radicals that I think are the most useful, the ones that are that have polar groups attached to them, so as to give them the right type of solubility and also reduce toxicity in living systems. Without further ado, I think I will let him take over and tell us about some of the latest developments in his research program. So, pleasure to have you here.
Thank you. So let me share my screen. Can you see it?
Yes, we do.
So thank you very much, Professor Rawal, for the introduction. So first, I would like to thank the O2M team for giving me the opportunity to present what we try to accomplish in my lab. So we are a fairly newly established lab. So I started my independent career in 2018 at West Virginia University School of Pharmacy. So we are also a member of the EPR Center at WVU. And our contribution to the field is mostly on the development of new EPR spin probes and spin label and hyperpolarizing agent for various applications. So we do the synthesis and the characterization in vitro and in vivo. And then we team up with biologists and clinicians for the application. So the previous speaker have already introduced the application of EPR to measure oxygen in vivo, but today I'm going to try to broaden a little bit the scope of what we can do in vivo with an EPR spectrometer, and I will present the recent progress from my lab in the development of spin probes to measure important parameters in vivo, such as, of course, oxygen, but also microviscosity, enzyme activity, and multifunctional probes that allow us to measure multiple parameters, such as pH, oxygen, and PI, which is inorganic phosphate. So today I'm going to focus mostly on the probe side, but not much on the applications. So for EPR, so for in vivo EPR, so we need, of course, a spin probe. So there is two main categories of spin probe use: particulate probe, which are insoluble, and soluble spin probes. And as our main goal is to do imaging, so we prefer to use soluble spin probes because they have the ability to diffuse into tissue. And within this category, there is two main classes of radicals used. The first is nitroxide radicals that has been known since a very long time, and there are hundreds of structures out there, so that's a very rich field. But in our opinion, they have some limitations in vivo. So the first one is that most of them are reduced fast in vivo. They also have broad line widths that decrease the functional sensitivity and the signal-to-noise ratio. The second class of molecules is the triarylmethyl, or TAMs, or trityl in short, of this kind. So those ones are highly stable in vivo, and they also show very narrow line widths, meaning high signal-to-noise ratio and high functional sensitivity. Those molecules have been designed by these companies on Nicomet Innovation by the late 90s. And they basically redesigned the original triphenylmethyl radical discovered by Moses Gomberg more than 100 years ago. And the idea was to avoid all of these hyperfine splitting. You can see that this trityl radical has a very beautiful but complicated spectrum. And to provide water solubility, it also increased stability. So this one is, of course, not water soluble, and it's also not very stable. So in order to avoid all of the hyperfine splitting, so they substituted all of these hydrogens by atoms, by elements for which the natural abundant isotope has no spin. And when you look at all of the commonly occurring elements in organic molecules, so carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, and fluorine, so there is really only 3 of them that don't have any spin. So they came up with this kind of structure, and you can see that every atom that bears significant amount of the spin density is either an oxygen, a sulfur, or a carbon. And because of that, the spectrum is a single line spectrum. So the carboxylic acid brings some water solubility, and it's also a very conjugated and bulky structure, so that helps with the stability of the radical, of course. And those molecules have amazing stability, so they are inert toward most biologically relevant redox species. The half-life in human blood is more than 24 hours, and the OXO 63 or 71 is so stable that you can inject it in vivo, recover it from the bladder, purify it, and reuse it again. So this has been shown by the group in Chicago. And really the only metabolite that has been detected after in vivo injection is this quinone derivative. So it has been shown that those molecules react pretty fast with superoxide and peroxyl radical to give this quinone. I think the second order rate constant is 10 to the power of 3 or 4, depending on the particular structure. They also react slowly with oxygen. So if you have a solution of this family of trityl radicals and leave it at air, over weeks. So you have a steady decay of the EPR signal, and the solution will turn from green to brown because this quinone is actually purple. To be complete, so the group in Paris, the group of Jean-Luc Boucher, they studied the in vitro metabolism of these molecules, and they show that if you incubate those radicals with liver microsome and reducing agents, so NADPH, under anaerobic condition, the reduced compounds, or the triarylmethyl, can be isolated. If you have oxygen in the solution, is the quinone which is isolated. And with peroxidase and peroxide, the cation can be formed, which can react with water to get the quinone derivative. This cation can also react with some nucleophiles in solution, such as amino acid, to give an adduct. The nitro radical can also react with this family of trityl radical with pretty high rate constant. And this leads to a substitution of the carboxylate to a nitro group. And if you have enough of this nitro, you substitute the 3-carboxylic acid, and you still have paramagnetic compounds. So those are not paramagnetic. So this we lose a signal, but in this case, we don't lose a signal in EPR. The same is happening with sulfite, an ion radical that substitutes carboxylate to sulfonate. And again, if you have enough of them, kind of substitute the 3-carboxylic acid by 3-sulfonate. So those are in vitro experiments, but from my knowledge, the only in vivo metabolite that has been detected is this quinone derivative. So the first application of those molecules is, of course, EPR oximetry, and this has been covered in detail by a previous speaker, so I will be pretty fast. So the collision between oxygen and the probes result in an increase of the relaxation rate R1 or R2 of the probe, depending on the concentration of oxygen. And the increase of R2 can be seen on the EPR spectrum through a line broadening. So if you have a CW spectrometer, you can calibrate the line width versus the concentration of oxygen. So this is one calibration of one of our trityl. But if you are lucky and you have an EPR, a pulse EPR spectrometer, so you can calibrate the R1 or the R2. And it has been shown by Boris and colleagues, Boris Apple and colleagues, that it's actually more an independent measurement to actually calibrate the R1 instead of the R2, because the collision between the radical itself also results in an increase on the R1 or R2 of the probe, but the effect on the R1 is smaller, while the effect of oxygen is the same for R1 and R2. So using R1 kind of limit the interference through the probe concentration. So those are probably the first 4 most known molecules to do EPR oximetry. Those are all like single line EPR spectrum, and the simplest structure is the so-called Finland trityl. It has a line width of 85 milligauss. So if you compare this with a nitroxide of, typical nitroxide has a line width of about 1 gauss. So that's significant increase in signal intensity. So the deuterated Finland trityl has line width even smaller. This is number from GE, so if you want to buy it directly from them. So the synthetic protocol has been published by the group of Driesschaert in 2002. So those are not pretty easy to make, but they have some toxicity issue. So on the other hand, OXO63 and the deuterated OXO63, that's also called OXO71, so are very biocompatible. So the line width is a little bit broader. So in the literature, the line width of OXO71 is reported to be 80 milligauss, but we actually found out that this corresponds to about 80–85% of deuteration of this position. So we managed to improve a little bit the deuteration, and we, in our case, we get line widths of 60–65 milligauss depending on the batch. And for DNP, so those molecules are also used for DNP and for polarization of pyruvic acid in human clinical trial. They actually use a derivative of OXO63, which has been methylated on the alcohol. And I guess the reason is because, so when you polarize pyruvic acid with oxo-63, it's very difficult to separate these two molecules very fast. So this one is more lipophilic, so it's possible to precipitate and then filter it out fast before injection of the pyruvic acid. But I don't think this one has been used for EPR oximetry. So as I say, the molecule, the synthesis of phenyltretine has been published almost 20 years ago now by the group of Driesschaert. That's a pretty important paper in the field. It's really jumpstart the synthesis of phenyltretyl-based spin probes and spin label for various EPR applications. After that, many publication has been, many improved synthesis has been published, including by us, but those are small iteration, a small improvement of the original synthesis. So now in my lab, we can make this compound, very easily. In 2 weeks, we make batches of 50 grams without any problem. And really, we are only limited by the size of the flask that we have in the labs with proper industrial equipment. We can make kilogram of this molecule without any problem. But these molecules have actually two big problems. So if you look at the structure, so the carboxylic acid is pretty polar. That's why the molecule is water soluble, but the rest of the molecule is very lipophilic. And because of that, it binds blood biomacromolecules such as albumin. And when it binds albumin, the correction time of the molecule changes, and this leads to broadening of the EPR line. And when we record the signal in the spectrum with low modulation and low power to look at the single, the narrow line, so we basically lose the fraction which is bound to albumin. This is what's shown here, is when you titrate with albumin, you have a decay of the EPR signal intensity. And this molecule is also highly toxic upon systemic delivery. So if you inject this molecule in a mouse, in large quantity, it dies in a few minutes. But it has been used upon intra-tissue injection, and it seems to be well tolerated, but the dose is like many order of magnitude lower than what's required for systemic delivery. So on the other hand, oxo-63 and 71, so they are more hydrophilic, so they have this 12 additional alcohol. And because of that, they don't bind albumin, and they have very high biocompatibility. So you can inject like 50 milligram plus to a mouse. The whole mouse turns green, but feels great. But it was done in the field to be very difficult to synthesize, and everybody we have talked with telling us that it was almost impossible to make. So we didn't try to reproduce the mistake of others. So we decided to develop alternative structure of this oxo-63 of 71. And basically what we needed is more hydrophilic structures to prevent interaction with albumin. And we made many derivatives. So these are two examples. So many derivatives of phenyltretyl with higher hydrophilicity to prevent albumin binding. So this is, for example, pegylated phenyltretyl. So another solution was to graft phenyltretyl to a hydrophilic dextran biopolymer. So those two options are great. So that actually resolved the problem of binding with albumin. But to be honest, they are still inferior to oxo-63 and oxo-71 in terms of high water solubility, biocompatibility, or ability of oxo-63 to diffuse very fast in the extracellular space. And probably one of the nicest structure that we developed in this road was this trisulfonated phenyltretyl derivative. And that's serendipity. So we kind of discover that sulfite in iron radical can actually substitute the carboxylate to sulfonate. And if you have enough of that, you just replace the tricarboxylate by trisulfonate. So we can follow this reaction by EPR. So when we mix, so this is the spectrum of phenyltretyl. So if we mix this with sodium sulfite and potassium dichromate, you can see a progressive conversion of the phenyltretyl to the mono di, and finally the trisulfonated tretyl, and they have a slightly different J factor. That's why we can resolve them. this is the study of the interaction with albumin. So DFT, if you mix 50 micromolar of DFT with 50 micromolar of bovine serum albumin, so you basically lose 80% of your signal. So this is because you only have 20% of the treated, which is unbound. And of course, more you put albumin, and the more you lose your signal. So with OXO63, I mean, you can put as much as albumin as you want. There is no effect on EPR spectrum whatsoever. So for this molecule, it's more hydrophilic and it doesn't have tendency to aggregate at high concentration, doesn't precipitate at low pH, but still binding albumin. Less than Finland treated, but still. So if you add one equivalent of BSA, we lose 20% of signal. So it's already better than 80, but still. And it's also toxic upon systemic delivery. So that's not the ultimate solution for in vivo, but for some in vitro application, it's pretty nice molecule, especially the line width is even more narrow than for Finland treated. And for my knowledge, that's the narrowest line width ever measured with the treated radical. It's actually pretty difficult to measure this. It's at the limit of what we can measure with our EPR spectrometer. So we need a very stable frequency and very stable field. As we could not find better alternative to this OXO63 and OXO71, so we decided to try to make it. And so as I know that most of you are not organic chemists, so I will not bother you with the synthesis of this molecule, but so Martin and Justin in my lab to spend some time to go through all of this synthesis and optimize it, and we finally succeeded in 2019. Since we made some improvement, especially to produce it in larger scale, and hopefully we'll be able to publish it at some point. So the property matches the literature, except that the line width, it's actually smaller than what has been reported, and this is because of the higher deuteration in this site. So now that we can do OXO63 and OXO71, so it opens the opportunity to make derivative, and one of the problem of OXO71 is the broadening of the line due to the concentration. So we are trying, okay, can we try to improve this, to solve this issue? And the idea was to replace the alcohol by carboxylate. And I did not write here the whole structure because it's not still ready to publish it. But the idea is to make a molecule which has more charges, and this would prevent collision of the radicals, and it would maybe decrease the broadening to the concentration. And this is the broadening to the concentration for OXO71 in red. So every millimolar of the probe, we increase the line width by about 7 milligauss, and for this molecule, it's actually a bit more than two times less. But it's also decreased sensitivity to oxygen. So sensitivity to oxygen for OXO71, it's 0.5 milligauss per millimeter of mercury of oxygen. So for this molecule, it's a little bit less than 2 times. And we were first a little bit disappointed, but then found out that it's actually not a bad thing, because the sensitivity to oxygen is actually more than enough. So we can measure oxygen with an accuracy of 1-2 millimeter of mercury. And I don't really think that we need that in vivo. And as you broaden the line, you actually decrease the signal to noise, and then you start to have a noisy spectrum. So having lower sensitivity, we actually decrease less the signal intensity at high value of oxygen. So then, turns out that the fit is actually better because we have higher signal to noise ratio. That's actually, I think, a good thing to have a little bit lower sensitivity to oxygen. So I would be very interested to look at the influence of oxygen and concentration on the T1 and T2 of this probe. It's something that we don't have the expertise in my lab. So if somebody would be interested to collaborate with us, we would be very, very happy. Okay, so that's second application of treated radical for in vivo EPR I want to discuss is the development of treated that are sensitive to molecular tumbling. So most of the EPR spectrum of treated are, they almost don't have any sensitivity to molecular tumbling or to the viscosity of the solution. And this is because one, they usually have a very small anisotropy of the G factor. And two, they usually have a very small, no hyperfine splitting or very small hyperfine splitting. And I think this is the biggest hyperfine splitting reported in the literature for this derivative that has hyperfine splitting of 3 Gauss. It means that even if the hyperfine splitting is not, has an anisotropy, the molecule doesn't have to tumble very fast to average this anisotropy and get a perfectly isotropic spectrum. But the effect on the mobility on the spectrum of a nitroxide radical has been one of their one of their success, and especially for side-directing spin labeling to measure viscosity of a solution. So we were wondering, can we develop a trityl that would be as sensitive as a nitroxide with the advantage of the stability of the trityl radicals, and working on the G factor seems to be very difficult because we would have to redesign the whole molecule. But so the idea was to try to get the biggest possible hyperfine splitting with a strong anisotropy, and all of the answer was actually already out there. So if you look at the EPR spectrum of a trityl radical, so if you just ignore this gigantic EPR line in the middle and you look at the baseline, so you can see multiple small lines that actually correspond to the C13 satellite at 1.1% natural abundance. And of course, closer you go to the central carbon and bigger is the hyperfine splitting, and this is because the spin density is bigger. And if you actually count all of these lines, so you can actually see that it almost makes almost 30% of the signal. So we can kind of lose 30% of the signal with C13, but that's pretty difficult to solve. And all of the credit goes to this paper from Michael Bowman in collaboration with Howard Halpern in 2005. So they actually measure with natural abundance Finland trityl all of these C13 lines in solid state and in liquid state, and they measure the gigantic hyperfine splitting from the carbon in the center, so the one that bears most of the spin density. So they measure 57.2 Gauss for the A parallel and 7.3 Gauss for the IA perpendicular. That was actually not measured, that was estimated based on the A parallel and the hyperfine splitting in isotropic in solution. But you can see that there is a strong anisotropy, and it's also a very big hyperfine splitting. So then we, as we are chemists, so we made this molecule, and that's luckily for us to label this this carbon with a C13. It's actually the most cost-effective carbon to label. It's pretty easy, so the same synthesis. We just use one C13 reagent. So Martin did that, and this is the EPR spectrum. Of course, it's a doublet because of coupling with the C13 with this gigantic hyperfine splitting of 23.3 Gauss. So this is in PBS, at room temperature. What is striking is, so we still have 1% of this C12 because we use a 99% labeled reagent, so not 100%. So we still have 1% of the C12 in the middle, and you can see that the line width between this and this are very different. So we have about 30 milligauss for deuterated Finland trityl, while those two lines are like 500, 600 milligauss, so more than 10 times bigger. This is because even at room temperature in low viscosity media, so the probe tumbled not fast enough to average the anisotropy, and that leads to borderline. And of course, if you prepare a solution not in water but in glycerol, so solvent which is highly viscous, so the tumbling rate is decreased, and then you get this beautiful rigid spectrum when you can now measure the AZ and AX, which is equal to AY because there is an axial symmetry to the molecule. And we got a lot of help from the group in Derwent, from Sandra and Gareth Eaton. So I'm not sure we will be able to do it without them. So thank you very much. And of course, instead of doing 100%, 90% glycerol, you kind of increase progressively quantity of glycerol to increase the viscosity. The viscosity of solution of glycerol are known. Then you go progressively from the doublet to this rigid spectrum, and using spectral simulation, so we can determine the correlation time. Then, of course, we can plug the correlation time determined by spectral simulation with viscosity, and then we can easily measure the viscosity of the solution using this technique. So if you look at the effect, so after 45% of glycerols, we start to have more like a rigid spectrum, but before that, so the effect is essentially a line broadening. So for in vivo application, which is our target later, so it may actually be easier just to use the line width as an empirical parameter to measure viscosity. And you can see that below 45% of glycerol, which corresponds to a viscosity of 6 centipoise. So we have a straight line and a gigantic sensitivity of 820 milligauss per CP, which is bigger than for nitroxide X band. So this is the same effect of oxygen, but the effect is like way bigger. So the sensitivity to oxygen, it's 0.5, 0.7 milligauss per CP. So we can neglect the effect of oxygen. This is way bigger. And of course, we already know that phenol tritine binds albumin, but with this molecule, it's even more clear. So if you mix phenol trityl with this, I think was bovine serum albumin, so we get this very nice spectrum in black. So we can clearly see two components. So these doublets that corresponds to the molecule rotating pretty fast. So this is the trityl which is not bind to albumin, and then you have this big rigid component that correspond to the trityl which is immobilized on the protein. So we can fit all of this, and we can extract the correlation time of the trityl which is bound to albumin, and we get value which is very close to the correlation time of measure to albumin with using like different techniques. And of course, I mean, our goal is to do in vivo. So there was some interesting paper out there like showing variation of viscosity for various diseases and as the disease progress, and including in cancer. So we want to use this technology to measure viscosity in vivo, but we cannot do it with phenol trityl. So I think Martin finished last week the synthesis of OXO 71 bearing the carbon C13 in the middle. So we'll be able to measure viscosity in vivo upon systemic delivery. So the third application that I would like to share with you, that's also a fairly new project, is how can we image enzyme activity directly in vivo. So there are many biochemical techniques to measure the activity of an enzyme ex vivo or in vitro, but to do it directly in vivo, that's pretty complicated thing to do. So we were wondering, okay, can we design a spin probe whose spectrum would be affected by the activity of an enzyme? And this is the first example that we did, and it's not perfect, but it's the first one. So we did this nitroxide bearing this phosphate, and this nitroxide is actually a substrate of alkaline phosphatase. So when the phosphatase does its job, so cleaving the phosphate, it releases the enol that undergo a keto-enol tautomerization, and we hope that the change in conformation of the cycle would have an effect on the hyperfine splitting with the nitrogen. So this is the spectrum of the substrate, and you can see that as soon as we add some alkaline phosphatase, so we can see the appearance of a second triplet. So the resolution is not great, but you can clearly see that there is a second triplet appearing, and if you zoom on this low field component and follow over time, you can see a decay of the enzymatic substrate and increase of the enzymatic product. And of course, to have this resolution, we had to use this fully deuterated nitroxide, otherwise we just have two broad lines and there is absolutely no resolution. So we were able to image this transformation. That's collaboration with the group of Mark Zetlin at our EPR center. So he has developed this amazing rapid scan imaging system. So this is spectral spatial imaging. So those are imaging over time. So in A, it's imaging of the substrate, in B, it's imaging of the product. So we follow this transformation in vitro in 3D printed phantom with bearing like three wells. In this well, we place the enzymatic substrate, so this molecule, no enzyme. On this one, we place the product, so this one, no enzyme. And on this one, we place the substrate plus the enzyme. And you can see when we look at the enzymatic substrate, so at the well that has the substrate but no enzyme, so we have constant intensity of the image. On the well that has the product, so we don't have any intensity because we are looking at the substrate. And on the well that has the enzymatic reaction, you can see that over time the intensity decreases. So we can plot the intensity over time, and you can see a nice decay of the substrate. And of course, this is concomitant with an increase of intensity of the product that you can see the product appearing when you look at the product. And of course, the sum of substrate and product is fairly stable. But of course, for our goal is always to do in vivo measurement and nitroxide. I mean, this molecule would be reduced in few seconds, I think, and the resolution is not great. So we are now working on treated radicals. So we got recently an R21 from NIH to develop treated radicals that are sensitive to enzyme activity and with the benefit of high stability of treated, and also narrow line width. It means the resolution of the peaks would be much better. So we've already got some interesting point data, but it's a little bit too early for me to present this now. And finally, the last application of treated spin probe for in vivo EPR I want to discuss is something that I started a long time ago. So I was a PhD student in Bernard Gallet's laboratory back in Belgium. So of course we were following the work of Valery Kramsov on development of nitroxide radical sensitive to pH. At this time, he was also starting to develop treated radical sensitive to pH. But as an organic chemist, it was pretty easy to understand that phosphonic acid was the best function to measure pH using a treated radical, because RL phosphonic acid have 2 pKa, so one of 2, which is not very interesting for biological application, but one close to 7, which is of course perfect for biological application. So we made this, so we substituted the 3 carboxylic acid of phenol treated with 3 phosphonic acid. This gives quartet because of the spin of phosphorus, which is 1/2. And of course, the hyperfine splitting is function of the ionic state of the phosphorus. So you can calibrate the hyperfine splitting with the pH. Then you can use this calibration to measure pH in vivo. So that's what we did. This molecule is not perfect because there are 3 phosphonic acids and they all have slightly different pKa. So the 3 phosphonic acid has pKa close to 7, but they are not exactly 7. They are not all the same. So it means that at pH close to 7, we actually have multiple ionic species that are not resolved. So they are on the lines. That means that it makes very difficult to measure both pH and oxygen at the same time because of this overlap of multiple ionic species. And at this time, it was already pretty clear that the molecule that we needed was not a substitution of the 3 carboxylic acid by phosphonic acid, but only 1 that would decrease the number of ionic species. But that was more difficult to make at the time. But of course, when the group of Valery Kramsov, they saw our work, they immediately started to work on that, and they were faster than us to publish this monophosphonated phenol treated. And of course, now if you have only 1 phosphonic acid, so you don't get a quartet, but you get doublet. So at pH 9, we have only this ionic species. So with this doublet, and at pH 5.5, we have only this ionic species, which again, a doublet, but with a different hyperfine splitting. So when the pH is close to pKa, you get the 2 ionic species. And then you can understand easily that if we know the pKa, just measuring the ratio of these 2 peaks, we can extract the pH. Still, the line width reports the oxygen. And they actually also show that inorganic phosphate modulates the rate of exchange between these two ionic species, and then we can simulate this on the EPR spectrum. So I will come to that in a minute. But their problem is they were not able to make more than a few tenths of milligram of this compound. So Valerie offered me a position in his lab, and we ended up working together back in 2014, and we are still now collaborating on this project. So now we call this molecule HOPE for its ability to measure pH, oxygen, pI. Those molecules, as they are charged and big, so they don't enter cells. Same for OXO71, so it's extracellular measurement. So that's why we measure HOPE. And this molecule actually allowed us to identify, and the work of André Bobko in the lab of Valerie, identify inorganic phosphate as a marker of tumor progression. And if you look, there is not many techniques to measure inorganic phosphate directly in vivo. It's something which is very difficult to do. But of course, this molecule has the same problem as phenylanthritol, so it's lipophilic. It binds albumin. So this is a zoom on the, I think, low field component. So if you add some albumin, you just lose EPR intensity, and also toxic upon systemic delivery. But so we, on daily basis, we inject this directly IT, so intra-tumor injection, and it doesn't seem to be toxic at all. But now that we have access of OXO73 and OXO71, so Teresa in my lab made this variant of monophosphonated OXO71, so we call it HOPE71. So the spectrum is basically the same. Resolution is a little bit lower because of a little bit broader line width, but still it's good enough. So those are the calibration. So we usually don't need to record the whole spectrum, so we can only focus on one component. So this is the effect on the pH, so at more acidic pH, so we have the acidic ionic species, and as we go more basic pH, we progressively convert to the basic species. So you can build a titration curve that allows to extract the pKa, which is 7.07, which is of course perfect for biological application. So the oxygen broadens the line. You can calibrate the sensitivity, so that's exactly the same as for OXO71. PI is a little bit more complicated. So PI modulate the exchange rate between the ionic species, and we can actually simulate this effect. So we can, with one single EPR spectrum, extract these three parameters in vivo. But what we found, it's pretty difficult to, so we used to fit this with a MATLAB algorithm, but it's pretty difficult to convince biologists to fit a spectrum with MATLAB. So Teresa used the best of her time during the lockdown so she built a standalone application that allows to fit automatically the spectrum of this kind of molecule. So the only thing that we need to do now is to load the spectrum from the L-band spectrometer and press simulate, and it gives automatically the value of pH, of oxygen, and pI. And so that was the problem with HOPE, that's bind albumin, and this new compound, so we can add a lot of albumin. There is no effect on the EPR spectrum. So with 200 micromolar of BSA, we lose 80% of the signal here. Here we don't lose anything. That's amazing, and it's also compatible with IV, so intravenous delivery. So we started some experiment recently, so on breast cancer model with mouse model that developed spontaneous tumor. So now we do retroorbital injection because it's a little bit easier than tail vein, and the molecule seems to be very well tolerated. So this is one example of spectrum that we got in vivo with using this mouse model. And again, you just need to load the file, press fitting, and we get automatically these parameters that can be done by any biologist. Okay, that's the synthesis, and I'm not sure if there were many organic chemists in the lab, but the way we do it, so we just oxidize this radical into cation using an oxidant, water-soluble oxidant. So we do the reaction in water, add phosphate that actually add on the para position, and lead to an oxidative decarboxylation and leads to this monophosphonated compound that we need to deprotect. So one thing I want to say before finishing, it's for some reason, each time I send a grant to NIH, I have at least one reviewer that says that it's very difficult to get the hands on those treatiles. So we are more than happy to share every molecule that we have made. So we have a website, so we even have like a category with all of the molecules that we have. So if you are interested, just send me an email, and if we have it in the lab, we can send you a sample. So we don't do any hoarding of treated radical. So most of the work that I have presented today was done by Martin, Teresa, and Justin. So those are new addition to the lab. And of course, I'm very fortunate to be part of this EPR center at West Virginia University, established by Valery Kramsov in 2015 now. So he has been my mentor as a postdoc, and now we are collaborating on various projects. Big thanks to Sandra and Garrett Eatons and their group for their help with the C13 compound. And those are other collaborations that I did not spoke about today. And of course, thank you for NIH for their financial support, and thank you very much for your attention. So we can take some questions if you have any.
Okay, very good. Thank you. Please join me in congratulating Benoit for a very nice talk.
Thank you.
So we will open up the presentation, the floor for questions. I noticed that in the chat, there is a question from Professor Bernard Gallet. He says, bonjour, Benoit, great job. What is your explanation for the difference in sensitivity to oxygen of the two soluble trittles that you showed? If it is just a matter of frequency of collision with oxygen, I would not expect this for a same partial oxygen pressure. This could be different for oxygen concentration. Could you clarify the point? Thank you.
Yeah, so the sensitivity of oxygen, where is this stuff? So basically depends on the frequency of the collision between the radical and oxygen. So this is described by the Smolensky equation. That depends on the concentration of oxygen, but there is also a parameter which is the efficacy of the collision. And I think that's the parameter which is important in this case. If you look at the calibration for Finland treated, it's even actually higher. So it's like 0.7, if I remember correctly. So for OXO 63, 0.5. And actually it's completely correlated with the bulkiness of the molecule. So more bulky it is, I think more the collision is less efficient. I mean, less the collision is efficient, and that decreases the sensitivity to oxygen. That's my explanation.
So I'm very interested, we are very much interested in this SOX71 because with low sensitivity, it might be better for us when we're looking into tissue engineering area, high oxygen area. So yeah, I would like to get some material and check T1, T2. As you said, we'd like to kind of characterize it at 25 millitesla.
Yeah, I mean, we can, I mean, I would be happy if you.
Yeah, definitely.
I mean, that's a new compound. So we did the first injection, I think, last week. So the mouse survived. So that's already a good sign. So we got an EPR image. But I mean, I don't know about toxicity, if it's less or more toxic than OXO 71. I would assume that it would be probably similar because, I mean, it's a very hydrophilic molecule. It's not cell permeable. That's what makes OXO 71 very biocompatible, in my opinion. So I would assume that this molecule is pretty biocompatible. So Teresa is actually working on that. So she's doing some cell viability assay measurement. So we will start some pharmacokinetic pretty soon. We will know more at some point.
Sure, sure. So feel free to send some to us and we can look at it. Thank you.
So Dr. Gianpaolo Vallarini has a question, and rather than my reading it, maybe it would be good for the attendees to meet Professor Driesschaert and ask the question. So Gianpaolo, maybe you can ask your question.
Okay. Yeah, sorry. I thought it was easier this way. So thank you for the seminar. It was very interesting. I have a couple of questions, mostly related to the biological activity of these compounds. One is if you can comment on the if it's known the reason of the high toxicity for systemically administered Finland Trito. And the second curiosity that I have is if the brain penetrability of triarylmethyl radicals is being studied, and if we know how they cross the blood-brain barrier and such.
Okay, so for your first question about toxicity, to be honest, I don't know. I don't think it has been investigated, at least it hasn't been published. So it's binding albumin. I'm not sure if it's what causes the toxicity. So I would not assume that it's the only reason, but that's highly toxic. Maybe there is some like solubility issue. Maybe at high concentration it may aggregate with cell membrane and maybe clog some capillaries. I don't know.
Yeah, I was thinking maybe would it be the tendency of form aggregates and precipitates to
that's possible.
Maybe drive the toxicity.
That's completely possible. I don't know if somebody, maybe people in Chicago, they know more. I don't know. So for your second question, those molecules are big, hydrophilic, so they don't cross the blood-brain barrier.
Yeah.
I mean, there is no chance. I mean, there's an artificial way to do it. I mean, you can, I think mannitol can open it up a little bit.
I believe, excuse me if I interrupt you, from what I understood from the beginning of your seminar today, you are trying to develop even more hydrophilic compounds because you get more finer line weight and sensitivity and better solubility. So that's the opposite of going into the direction of designing brain penetrable compounds. How would one how could one design effective spin probes, but with a lower hydrophilicity and able to cross the blood-brain barrier, if that's something you can even think of?
Yeah, I'm not sure triarylmethyl are the best molecule for that. I think some nitroxide have been designed that can partially penetrate the blood-brain barrier. But that's a difficult thing to do. I mean, those molecules are big,
yeah.
and I mean that's the role of the blood-brain barrier, is just to keep everything out.
Mm-hmm.
Yeah, I'm not sure those molecules are the way to go.
Yeah. Thank you.
Mm-hmm.
So I had a question about the compound that Marignani asked about, the oxidized form of OX63, I guess.
Mm-hmm.
Yeah.
Okay.
Do you see any deuterium leaching under physiological conditions from enolization and reprotonation?
I mean, here I did not put the right structure. So there is this dotted bond. So this carboxylic acid is not directly connected here because I know that this seminar will be put online.
I see.
Ready to publish it yet?
I see.
So the carboxylic acid are a little bit further apart.
Further apart. Okay.
So then we don't see any.
Yeah.
This molecule would be actually pretty nice to make, but we could not make it that close. Yeah.
So the idea of putting a C13 at the trityl carbon, the central carbon, that was very interesting. Would you expect any benefit in having that at the thioketal carbon?
It would just decrease the effect on tumbling of the radical. So the most sensitive would be to put it on carbon, and So I don't probably.
No.
So if we would put it here, so the coupling is pretty. It's actually very small. It's like
Yeah.
Also, I mean, we would get this doublet, and it would be pretty much insensitive to molecular tumbling. I don't think it's.
Yeah.
I don't see any application right now.
Okay.
Yeah, this one would be also easy to label with the C13.
So with the extensive experience you have in the chemical synthesis of these compounds, what would you say is the most difficult challenge still in the synthesis for either the Finland compound or the higher oxidized versions? What is the most challenging step?
I mean, for us, the biggest like technical problem that we have is to make this molecule, and there's nothing to do with the chemistry. It's just that we need to use like huge quantity of triarylmethyl thiol, and we can take any precaution. It smells bad in the whole building every single time, and that's actually a big problem because I was forbidden to do it in Belgium. I was forbidden to do it at Columbus, and now we are very close to be forbidden to do it here at WVU. So I don't know how we can. There was another paper that's been published recently, an alternative of using directly like triarylmethyl thiol. So we tried that, but it's better, but still smell on very large scale. It's still very smelly. So otherwise, I mean, it's pretty simple organic chemistry. I mean, there is nothing. Yeah. I mean, it's everything is standard. So we found for the synthesis of OXO63, it was really only two steps that were kind of challenging. It was making the trityl from the monomer, and actually adding this carbonyl. And maybe you can there is something that I still don't understand those days. So if you treat this molecule with sec-butyl lithium intermediate and you add CO2, you actually get a pretty good carbonylation. But if you put any other electrophile, like a chloroformate or a carbonate, there is almost no yield at all. And this I cannot explain why CO2 is working that great, one other electrophile don't work at all. I mean, I cannot not understand that. And I think that's why, I mean, many people tried before and they were trying to put directly this, trying to get the ester directly, not using CO2, but using diethyl carbonate or dimethyl carbonate. But that doesn't work. I'm not sure why. Because I mean, aryl lithium on the carbonate would react like very well. Yeah, I'm not sure. And the solvent, I mean, it was, I mean, the solvent were very important and the temperature, it's very fine line at which those reaction actually works.
Yeah, it's very subtle chemistry and requires great control over all aspects of the reaction.